Thank you very much. It's a real pleasure to be here, and I thank you for the invitation. Give me just 1 second. I'm going to share my screen and get started here. Okay, should be all, let's see. Okay, it should be set now. Anyway, again, thank you all for the opportunity to come and talk to you. So I was asked to obviously talk a little bit about the history of the MBL, and honestly, probably there are many of you that know much more about the history of the MBL than I do. It's an amazing institution that was founded in 1888, and as Debbie said, was the second scientific institution here in the area. So what I thought I would do is review parts of the history and then tell you how that basically informs the directions that we're going, and then give you an update on some of the directions that we're headed. So I think all of you know that we're located here in lovely Woods Hole, and it's an amazing location for doing research. So a little bit of the history then. So it really, I think, I was going a lot on some of the wonderful documents that have been collected by the MBL library that describe the history of the institution, and many of the great writings that have been put out there about the history of the MBL and of Woods Hole and the great scientific tradition of all the institutions in this area. So I think it dates really back to 1871 with the establishment of the United States Department Commerce Bureau of Fisheries. And the original director of that, Spencer Fullerton, established a permanent research station for the Fish Commission here in Woods Hole. But the actual beginning of the sort of nascent MBL really begins in 1887, where the Women's Education Association appointed a committee of 12 members to perfect plans for the organization of a permanent seaside laboratory that would combine research and instruction. And this led then in 1888 to the incorporation of the Marine Biological Laboratory. Its first director was an embryologist, Charles Otis Whitman, who at that time was at Clark University, but was about to join the University of Chicago. And it was Whitman that was appointed as the first director of the MBL. And so that's our beginnings all the way back in 1888. Now, the thing about the establishment of the MBL and other institutions, so you say, well, why this area of Cape, why Cape Cod, why this particular area? And it really has to do with some of the interesting biology that's out here. And the reason that it's out here is the merging of warm and cold currents. And so you have both populations of organisms that come from colder water and warmer water, and you also have upwellings of nutrient-rich waters. And so this leads to an incredible level of biodiversity in this area. And that is one of those things that I think really attracted some of the early scientists to develop a marine lab in this particular location. So if you go into the water around here, you might say, well, it's not like a coral, a beautiful tropical coral reef that I would think of as the ideal place for a marine lab. But in fact, the reason that the water looks the way it does is that, in fact, it is actually very nutrient rich. It is full of biodiversity. And so there's an incredible array of organisms that we find in the waters around here. Some of the things are big things that you all are familiar with, like, whales or striped bass. But it also teems with much smaller organisms of all different kinds of phyla that really attracted a lot of early investigators to thinking about all of the different ways they could use these marine organisms for research. So it began really as a summer-only institute, but it grew very rapidly. And again, it had an emphasis both on education and research. And so it started off in the first summer with just 17 students, and then it grew very quickly to over 100 students coming here. And one of the notable things is that women were well represented from the start, and that was actually a great thing, and has to do with the way it was founded, and it's a tradition that we've, I think we've been very happy to continue. The first summer course that was offered at MBL was in invertebrate zoology. Interestingly enough, it's morphed into a course that's now called Neural Systems and Behavior, but it was soon complemented by quite a number of other courses, including embryology, physiology, and courses on microscopes. Now, embryology is near and dear to my heart because that's the thing that brought me here. Well, actually, I have to say that the molecular evolution course was the first course I was invited to, but 20 years ago, I started teaching in the embryology course. And so this will be my 21st summer being around the MBL, helping to teach. And those courses have been, an incredibly vital part of the MBL. So they were a number of research areas that were followed by the scientists that visited in the summertime. And they explored such things as the interplay of evolution and development and the fine structure of cells. There were quite a number of, really luminary scientists that spent time here. And it's a huge long list of amazing people that have been associated with the MBL. But just to highlight one of the early, investigators here, Thomas Hunt Morgan would, pack up and come here from Columbia in the summers. He did that for decades. And, he pioneered work in the fruit fly, Drosophila melanogaster, and went on to win a Nobel Prize for that work. And he's just one of many, many examples of amazing scientists that have credited the environment of the MBL for being able to do the science that they do. So fast forward now, quite a long time. And so in July of 2013, the MBL became an affiliate of the University of Chicago. Now, actually, there's a long history of ties to the University of Chicago. So as I mentioned, Whitman, the first director, was also at the University of Chicago. So several of the buildings at MBL are named after faculty of the University of Chicago. So there have been long ties, but those ties were in the past. The more recent ties really came in large part from Bob Zimmer, the president of the University of Chicago, who was very familiar with the MBL from his time at Brown University. And so it really was Bob Zimmer that really pushed forward the extraordinary value of the MBL to the university. And so we became affiliates in July of 2013. So as I mentioned, there's an incredible history of outstanding research that's been done at the MBL. So as we like to list at our website, there are 59 Nobel Prizes that have some sort of affiliation with the MBL. Now that varies quite a bit from people who are doing research here really intensively, and much of that, and some of those groundbreaking experiments were really done at the MBL, to people who were parts of courses, had taken courses here and things like that at the MBL. But these are all people that really had notable affiliation with the MBL over the years. And those Nobel Prizes and other, prizes and things like that have been awarded for a lot of very key discoveries in the area of biology. And so I list just a few here. So for example, early research that uncovered how impulses travel along nerves was done here at the MBL. Early pioneering work on using horseshoe crab blood to detect bacterial contamination, a technique that's still used to this day to test medical equipment. The discovery of the proteins that control cell division, both in normal development and in cancer. Much of those breakthrough experiments were done here at the MBL, and how molecules are transported within cells. A lot of that was pioneering work that was done here at the MBL. And so the list goes on and on of incredible discoveries. But in 1902, there really was a sort of a pivotal moment for the MBL in which it could have gone in a very different direction. So in 1902, MBL had become reasonably financially independent fairly quickly, but then as it grew, then it depended again on various streams of money. And there was a point where an affiliation or association with the Carnegie Institution of Washington was put forward. And actually, I have to tell you, the Carnegie Institution of Washington is something near and dear to my heart, because that's actually one of the places that I worked for quite a number of years before I started my first faculty position. But it was known for having pure research institutions scattered across the United States. And so there was a suggestion that the MBL actually become part of the Carnegie Institution and be strictly a research institution. Now, Whitman rallied support against this, and it's because to him, he felt that it was really important that MBL be both an institution of research and education. And fortunately for us, in terms of thinking about the two of those things, Whitman carried the day, and the MBL didn't associate back then with the Carnegie Institution and continued to be a place that valued both research and education. And I think this has really been a key to the success of the MBL. So I want to talk a little bit about the research. So here's an old picture from the start of the embryology course back in 1893. And so again, this is one of our longer running courses. And it's an amazing course that's produced quite a number of outstanding scientists. So for example, here's the class photo from 1979. And included in this picture is Tim Hunt, who in 2001 won a Nobel Prize, and Joan Ruderman, who went on to become the first female director of the MBL. And here's an even more recent slide, actually probably about 5 years old now, of teaching in the embryology course. And this continues to be the premier course in the world for people who are interested in developmental biology and embryology. So we offer actually a wide range of these advanced training courses in the summertime, about 21 of them, and you see them listed here. And they cover a wide variety of topics, from development and reproduction to cell physiology, microbiology, infectious disease, courses really focused on microscopy, and then a variety of courses in the field of neuroscience. So these courses are actually, though, designed for, first of all, they're in the summer, and they're designed for people who are graduate students or postdocs, and occasionally junior faculty. But they're designed for people who are, past their undergraduate career and are actually in research programs, again, as PhDs or as postdocs. And those are the students that take those courses. And they are, again, amazing courses. They are the courses that define these fields of biology and that everybody would like to get into. And these dominate our summer. So they take us from, we have about 250 people at the MBL in the middle of the winter, but our population rises to over 1,300 in the summertime because of not just these courses, but also all the visiting scientists that come and their students and everything that contribute to the environment. So one of the things that struck me in all the years that I've been teaching here is that we have these amazing resources and a fantastic building for teaching, but it remains largely unused during the rest of the year. And so one of the things that we've been doing at MBL is really trying to find ways to expand our education mission, to make greater use of all of those resources that we have. Now, the one undergraduate course that actually does have a nice long tradition at the MBL is the semester in environmental science course, which has been going on for over 2 decades, I guess, now, and is an undergraduate course that attracts really great students in the fall. But again, we've been thinking about how to expand all of that. So there are a couple of things that we've done. One is to launch a set of secondary or high school courses. And we got this started about a year before the pandemic shut us down. And we had a few schools that had come through. I think it went very well. We were offering courses in things like anatomy and development, the microbiome, gene editing, and so on. And for the most part, these were designed as 6-day courses where the students would actually stay here in residence. And so they were designed very much like our advanced training courses, really intense opportunities for the students to work in the lab and to be next to scientists and to learn cutting edge techniques for all sorts of areas. We were hoping to attract local schools. We were actually just about to have schools, a couple high schools from Cape Cod come to take the high school course when suddenly we were shut down by the pandemic. And so we are very excited to get back to this as soon as we can do it safely. So probably the earliest we can do that is next fall, but we hope to attract local schools to the program that we're able to offer. In addition, then we've expanded our offering of undergraduate. Oh, sorry. I'll just show you a little bit of the kinds of things that we allow these students to do. So this is a fly embryo, and this is a course that I helped to teach on anatomy and development, where we did a lot of advanced imaging. And so, this is a three-dimensional reconstruction of the nervous system of a fly embryo. And this is using incredibly sophisticated equipment. So a microscope that's probably valued at over $400,000, but we teach the high school students how to use this. I mean, we tell them not to break it, but the idea is that we get them to do sort of cutting edge science, and they love it. And it's a great opportunity for both for them and for us. It's a great teaching opportunity. And then in addition, now we've been expanding our undergraduate offerings. So I mentioned the SES course, which has already been in existence and running for a while. We also had September courses for UChicago undergraduates. So it's a 3-week long set of courses that have been running now for a couple of years. But the important expansion that we've made now is to start welcoming UChicago undergraduates during the non-summer months. So in the fall and the spring quarter. And we actually started off this fall. And so, of course, it was a challenge because of the restrictions caused by COVID-19, but we were able to do it. We were able to provide them a safe and I think really, really excellent educational opportunity for them. And this just shows one of the students, and again, some of the imaging that in a course that I did. And we are running classes in spring of 2021 that are in imaging, stem cells and regeneration, and microbiome studies. Okay, so back to the research. So as I mentioned, the sort of two pillars of the MBL are the education and the research. So research now at the MBL not only encompasses those visiting scientists that come in the summertime, but we have a long-standing now resident research program of outstanding scientists that work in a number of areas. So these just illustrate some of them. So we've had interest in many, throughout the history in biological discovery in marine organisms. A lot of that from our resident researchers is focused in the area of regeneration and tissue biology at the Bell Center. Then we have the Ecosystem Center that studies ecology and climate change that has been here and very strong at the MBL for many years. And then in the Bay Paul Center, there's been an increasing focus on the microbiome and its key interactions to our human health, the planet, and the ocean. And then there's always been a strong emphasis at the MBL in imaging and microscopy, and that helps to actually drive some of these other areas. And so what I want to focus on now are the two sort of newer research initiatives that I'm really pushing forward in the last, couple of years here at the MBL. And, we're continuing to grow our strengths in the microbiome research and in the ecology research and in all those areas. But these are two things that I've been focused on right now. And one is the development of new genetically tractable organisms. So again, the MBL has had a great tradition of working on a variety of marine organisms to make important biological discoveries. So for example, those discoveries I told you about first understanding how nerve impulses are carried was done in squid because they have these giant axons in them that were tractable for experimental study. But nowadays, one of the things that is really exciting about working on organisms is the ability to genetically modify them. So probably many of you have heard about the excitement of CRISPR-Cas9 genome editing. So this opens up a whole new world of studying interesting organisms and being able to do it in a much more experimental way of attacking them for molecular biology and genetics. And the other is to expand our resources in imaging and in microscopy and being able to see increasing levels of complexity in biological systems. So both of these are areas that MBL actually has played important roles in, but our hope is to take them to new levels of excellence for both our research and our education. So again, we've had a great history of some beautiful innovations in microscopy. So for example, Shinya Inoue, right, using polarized microscopy, was able to do some absolutely phenomenal experiments looking at living cells and understanding how chromosome segregation works. And so people before had done it in fixed tissue, and there was lots of arguments about, well, we're what may have been real or what may have been artifacts in those dead cells. But Chinya was able to do it in living cells and to show exactly what was going on. So we build upon that strong tradition that's existed at the MBL. So I won't go into great detail, but basically what we're doing in our imaging center now is really beginning to push forward at the boundaries of what limits what we can image in biological systems. So there's always limitation by how bright you can make something, how much light you need. So this is a trade-off. The more light you shine at the living cell, the more likely it is to die. So, but the better you can see with the more light you have. So how do you make that trade-off? Time, how long can you keep things alive under the microscope? Resolution, what's the smallest thing you can see? And what's the diversity of sort of organisms that you can image? So we've been doing a number of things that are designed to basically take this pyramid of what we can image and expand it to improve many of the ways that we do microscopy. So for example, in the last decade or so, been some incredible breakthroughs from physicists on going beyond what were thought to be the physical limits of resolution with a microscope. And so we're incorporating those into our current microscopes at the MBL, and different ways to actually increase the ability to see with less and less light. And so a variety of innovations that we're making in this area to be able to do that. And then the other one is to look at increasing the diversity of research organisms that we have. And again, with the idea of not just organisms that we've always collected and or will always be useful for researchers at the MBL, but to really develop ones where we can do genetic manipulations in them. And so why do we want to do that? Well, part of it is a long tradition at the MBL that we've been interested in evolutionary mechanisms and what it is that unites and makes different lineages of animals either similar or dissimilar. But there's also lots of amazing biology that's not well represented in our current major models. So if you read any, of most of the literature in biology, it's focused on a very small number of organisms, things like Drosophila melanogaster, the fruit fly, or C. elegans, the nematode worm, or mice. And there are good reasons for those things. They have incredible communities and they have incredible tools that have been developed in them, but they don't represent a lot of interesting biology, and they don't represent much of the diversity of life. So if you really want to study the diversity of life, the best place to go sample that is in the ocean, because that's where life started and that's where most of the diversity still lives. And there's incredible biology, from incredible examples of bioluminescence to one of the things that many MBL investigators study, which is regeneration. So none of our current major research models that people work on regenerate very well. But there are plenty of marine organisms that regenerate exceptionally well. And so those are one of the breakthroughs that we think can be made by focusing on some of these new research organisms and then making them genetically trackable. So one of the early organisms that the MBL has taken up as part of this new initiative in terms of making genetically trackable organisms are cephalopods. So probably some of you have read of some of the great work that's been going on in the MBL studying squid and cuttlefish and octopus. And so this just shows one of the animals that we work on called the Euprimnoberii. And a number of investigators led by Josh Rosenthal and Karen Crawford and Carrie Aldrich at the MBL have been able to genetically modify now, in this case, these are the squid, and they've knocked out a gene that contributes to the pigmentation. And so they've been able to make squid without pigmentation. And this is really more of a proof of principle experiment that they can now do these kinds of manipulations. So we're very excited by all of these kinds of areas of progress. We're excited to continue to grow the MBL, both in terms of its research and education, and I'm happy to take any questions. So thank you all very much.
Great. Thank you, Nipam. Anybody have questions? You can just put them in chat if you have them. Okay, no one has questions? I can't believe.
Yeah, there's a couple. I can start answering them. So I see them here in the chat. So we have a question, how do you sign up for a summer course? So the summer courses, like I said, many of them are directed at people who are in their graduate programs and postdocs, but there are also courses for science journalists and so on. And you can find all of that information at the MBL website. So if you go to mbl.edu and navigate to the education pane, you will see all of the courses listed. It provides all of the deadlines for the courses, how to apply. And, we are going to have an interesting time this summer still, because we still are dealing with the pandemic. So we will have courses going on, but they'll be at a limited capacity with fewer students and more spaced out, following all the state guidelines. But we are looking forward to that. But anyway, if you want to know more about the courses, all of the courses, you can go to our website and you can see the vast number of courses that we offer and how to apply for them. There's another question here. What does genetically tractable mean? So yeah, so what that means is that we can do genetic manipulation. So we can, for us, it means that we can knock out or insert genes. So we're very interested in, like when we look at regeneration, we can identify what we think are candidate genes that may allow regeneration. So for example, a gene that may be expressed in something like a lamprey during spinal cord regeneration that is expressed in lamprey, but not expressed in something like a human or a rat that doesn't, that isn't able to regrow its spinal cord. But the question is, we identify that, how do we functionally test that that's what it does in an animal like, a lamprey or some other marine organism? So what we need to be able to do is to manipulate that gene. And there are techniques to do that in things like mice and fruit flies and nematodes. But now especially with the advent of CRISPR-Cas9 genome editing, we can do it in almost any organism. So for example, my own lab works on a tiny little crustacean called Parhyella, and it's very easy for us now to go into that animal, knock out genes, see what the effect is on the animal, insert genes, see what the effect is on that animal. So it takes us to a whole new level of being able to do experimental manipulations. And then we have a question, it's great, and why have women always had a major role at the MBL? I'd like to say that, in part of it, it's because, we have this foundation from being founded by the Women's Association. So that obviously played a role, and they played a guiding role in the foundation of the institution. And I think we've always valued having diversity in our courses. Now, have we always been great at that? No, we're trying to do better. But I would say, and I take great pride, for example, in the embryology course, that in fact, we usually have more women than men in that course. One of the things, though, that we and other institutions are really striving for is to make sure that people that they continue in science, continue up the ladder, whether it be in academia or industry, and become the leaders that actually are, the presidents of universities and things like that. But MBL does really take great pride in having done, I think, a reasonable job, but we understand that we can do much better and are continuing. That's one of the things we're really focused on, is trying to be much more diverse and inclusive in everything we do. Okay. Where does the funding to be going for basic research? I guess I think that means, where is it coming from? So, the majority of our funding that supports our research long-term comes from the federal government. So National Science Foundation, National Institutes of Health, Department of Energy, and so on are the ones that provide the grants that continues to fund basic research. And a lot of that, you might ask, well, why does the National Institute of Health fund basic research? It's because most of those pioneering studies that have led to the breakthroughs that improve human health have started in other organisms. And so, it's the basic biology that needs to be done that leads the way for those discoveries. Let's see. How concerned are you about genetically modified organisms that escape and change the world around them? That's a great question. So we, for example, at the MBL, our genetically modified organisms, we're very careful to contain them, but we aren't actively engaged in creating any organism, I think, that could do any damage. We knock out genes, which usually actually leads to the death of the organism, in fact. And so these are not things that are going to go out there and change the environment. But nevertheless, we have to realize that there always is that possibility. So we are very careful about containing any of the genetically modified organisms that we make. But I should say that, in many ways, the nature is much more capable of making some very dangerous genetic modifications on its own. But it is a serious concern. And we've had the ability to make genetically modified organisms for a long time and have actually dealt with it quite well, we meaning the scientific community. Now we just have other organisms that we can do that, but we are conscious that these are not things we want to release into the wild. Let's see. So there have been many other novel models developed here, especially cnidarian models, Nematostella and Nemeopsis astrangia, but they are rarely celebrated. Let's celebrate them. Absolutely. So that's a great point. The MBL has really led the way in helping to sort of champion some of these animals. And they've been developed by scientists, not just the MBL, but kind of all over, especially at marine labs. And I think it is time, when we are making a concerted effort to educate the public about how important these organisms are and the incredible insights that have been made. So for example, these cyclin proteins that control the cell cycle, few students ever really learned that in fact they were discovered from experiments done at the MBL in sea urchins and surf clams. Right? They didn't start off as someone studying humans or even mice or anything like that. And it is important to celebrate those kinds of discoveries. Is private grants and development a future prospect? So I think that gets to sort of the idea of going after philanthropy as well. And we do that, and the MBL has a great track record of doing that. We often use philanthropy to get programs started. So, we go to donors and make a pitch that the federal government, we want them to eventually fund something that looks promising, but we have to get the preliminary data. And so philanthropy often ends up being a great way to do that. And the MBL is really fortunate to have many outstanding friends and alumni that have supported us in that way. Oh, maybe mention the Friday evening lectures for the summer. So yeah, we are, last summer we did all the Friday evening lectures remotely, but this summer we are going to continue with our great tradition of Friday evening lectures. We'll have kind of a hybrid approach. Lilly Auditorium can only hold about 70 people according to the current COVID-19 guidelines. So what will happen for the Friday evening lectures is there'll be a hybrid. We will allow some people in the auditorium, but that will mostly be the students and faculty here who are here for the courses, who are part of our COVID-19 testing program. And they'll be the in-person audience, but we have an exciting lineup that I think will all be posted soon, but you all will be able to join us virtually. And we were amazed at how many people joined in for our Friday evening lectures last summer, way more than could fit in Lilly Auditorium. So I think we've all learned. There's a lot of things not great about Zoom, but there's a lot of great things about Zoom. And one of them is appealing to really large audiences. So it's fantastic to think that we can have an audience bigger than would fit in Lilly. So, Lilly would hold about 500, and we had FELs that had like 700-800 people. We even had one of our lectures for one of our, the embryology course, have 800 people come to it. Normally we're in a room that can only hold 50. So anyway, we're excited by all those things too. Let's see. Part of the MBL history involves organizational marriages that didn't hold. University of Chicago is relatively new. Bump, Boston University, and Aquavet UPenn were here for many, many years. That's absolutely true. But in this case, those were alliances that only involved part of the institution. We are now in every way part of the University of Chicago. I mean, we are still an independent 501c in the state of Massachusetts, but we are integrated in with both the research and education now and missions. And so, I think we are very excited by that. We have a lot of work to do to make that association stronger and to make it more beneficial to both sides, but we do receive great benefit for it. So for example, by being part of a much larger institution, we can turn to them for all sorts of help. So when we need, more sophisticated advice on our IT needs or on, purchasing or getting insurance and things like that, it's great to be part of a large institution like the University of Chicago. Okay. Do scientists get to work under the do students get to work under the scientists and learn that way? Yeah, that's part of what we do. So we have internship programs and things like that for students. Even in our courses, our advanced training courses, we really encourage the students to work with the scientist faculty that are there in the courses to try new things. I mean, one of our favorite sayings is that one of the most important things our courses teach our students is how to fail. Because we tell them if you're not failing, you're not really trying. So, fail some more. And then when you succeed, you'll probably have something that's really amazing that you've managed to accomplish. And it's great to work with a scientist. I love teaching in the course, but I have to say it's exhausting keeping up with the students. So we often start at 9 in the morning and end at 3 or 4 in the morning. So, but we're driven by the students. We always tell the students, if you're here and want to work, we're here and want to help you. And so I think it actually works both ways. It's great fun for the students. Great fun for the faculty. So another question is, how does one launch a new initiative? Is it the Wild West, or must new ideas comport with strategic plan? Say, just take a random example, somebody wanted to begin a research thrust at the MBL on chaos and determinism. It's a great question. So, the two initiatives, the microscopy and the new research organisms initiative, were actually launched before I became director, but I was involved in both of them. So I was involved in discussions with the faculty here, the Whitman investigators in the University of Chicago, thinking about things that the MBL could do as new research initiatives. So a lot of thought has really gone into this, and we have external advisory committees that advise us in both of these initiatives. And we have very close partnerships with several foundations that are helping us with both of these initiatives. So it is possible to come and have discussions about other initiatives. But of course, there's a certain bandwidth that we have. We're focused on these two at the moment, but there is plenty of room to think of other ways that we can expand all that the MBL does in research and education. And so I always look forward to those kinds of conversations. There's a comment that Louis Agassiz, Edward Sylvester Morse all taught in the courses at Penakees Island for teachers in schools, forerunner for the MBL accounts for the women. That's probably also an important aspect of it. I mean, it's an interesting relationship with what went on at Penakees. It attracted some of the earliest people here, including the women that came here, and then, the Women's Association. There was a gap, though, between, I guess, my understanding of the history was there's a gap between the Penakees School and then the founding of MBL. Okay, we have a question about why did we change the seal? So as many of you saw, we announced that we've had a modification to our logo, and you saw it there at the beginning of my talk. But we actually still also, as you may have seen from my opening slide, retain the legacy seal. So the reason for the new logo was that in times nowadays, we need a logo that can be seen when it's quite small, especially for electronic formats. And so we wanted a simplified logo that also incorporated some of the newer things that we do at the MBL. So our legacy seal, which actually has a kind of complicated history as to how it became the seal, it's great for showing the diversity of marine organisms that we work on, but it lacked a nod to the amazing work we do in ecosystems research and the work that we now do in the modern field of molecular biology. So we felt that our new seal our new logo helped to do that. But don't worry, you will be able to always buy merchandise with a legacy seal on it, and you will always see us use both of those when they're appropriate. But the new one is nice for when we need, say, for example, a business card where you can see the logo really easily. So I hope we've gotten a lot of, I know that there have been mixed comments on the new logo. I think it's really great. We've gotten a lot of positive comments, and I hope you guys will enjoy it. Let's see. Traditionally, the MBL has been well integrated into the Woods Hole community, like with the Children's School of Science, the MBL Club, for kids, the periwinkle. And that's absolutely true. We hope to continue that. Obviously, for this summer, we have a lot of challenges for childcare, but that's very important to us, and we're working through exactly how we're going to offer it. We've just finished taking a poll amongst all of our visitors and our resident faculty about how they would like to see us, what sort of options we might be able to propose for childcare this summer. But those are important things. And it is great to be part of the incredible tradition of Woods Hole in this city and the 5 other scientific institutions. And I think it's part of what makes MBL and Woods Hole so great, is that we're part of a big science community, which is really wonderful. Okay, I think that's most of the questions that I think I can answer here. So I thank everybody for listening. And
Okay, well, thank you so much, Nipam. We really appreciated that. I've learned a lot. And just so you know, this has been recorded. We'll post it on our website, so you can watch it again. And we'll have another one next month on the Woodwell Climate Research Center with Philip Duffy and Heather Goldston. So keep posted about that. And thank you again, and thanks all for coming.
thanks, everyone.